What if a single gene holds the key to our linguistic secrets? In 1998, scientists studying a British family with severe speech impairments made a startling discovery. 3 generations couldn't form words properly or control complex mouth movements. The culprit, a single mutated gene called FOXP2. This clinical observation opened the door to a profound evolutionary mystery. When researchers compared this gene across species, they found something unexpected. Humans possess 2 amino acid substitutions not seen in any other mammal, changes that appeared suddenly in our timeline. Even more baffling, these same changes were present in Neanderthals, raising profound questions about where our language abilities truly originated. The discovery that changed everything. What if the key to humanity's greatest achievement was hidden in a single gene? Language, the ability that separates us from all other species on Earth, might come down to a tiny sequence of DNA that scientists only discovered through an extraordinary accident. In 1998, researchers in the UK were studying a family with a peculiar medical condition. Known scientifically as the K family, they presented doctors with a mystery that had persisted across 3 generations. Half the family members struggled with a severe speech disorder that made normal conversation nearly impossible. The affected individuals couldn't form words properly, had trouble understanding complex grammar, and struggled to make the precise mouth movements needed for clear speech. The K family's condition was unlike anything doctors had seen before. It wasn't caused by general cognitive impairment. The affected family members had normal intelligence. It wasn't a result of physical abnormalities in their vocal apparatus. The disorder followed a clear inheritance pattern. 50% chance of inheritance from an affected parent, the classic signature of a dominant gene mutation. Daily life for affected family members was challenging. Simple tasks like ordering food at restaurants became ordeals as servers struggled to understand their speech. Children in the family would often rely on gestures when words failed them. One teenage member reportedly carried written notes to communicate essential needs when her speech couldn't be understood. Their struggle extended beyond spoken language to written forms and even sign language, revealing a fundamental language processing problem. Linguist Myrna Gopnik, who first studied the family in the 1990s, recognized the scientific significance immediately. Here was potential evidence for a genetic foundation of language, something that could help answer long-standing questions about how humans evolved this unique ability. A team led by geneticist Anthony Monaco at Oxford University embarked on a painstaking search for the genetic cause. They collected DNA samples from family members, both affected and unaffected. Through linkage analysis, they traced the disorder to a region on chromosome 7, narrowing down the search from 3 billion base pairs in the human genome to a manageable section containing about 70 genes. The breakthrough came when they identified a specific mutation in one of these genes, a single letter change in the genetic code that disrupted a protein called FOXP2. In the affected family members, this mutation created a truncated, non-functional version of the protein. The researchers confirmed their finding by identifying the exact same mutation in all affected family members and none of the unaffected ones. The discovery made headlines worldwide as the first gene directly linked to speech and language ability in humans. But what exactly did FOXP2 do in the brain? Further research revealed it functions as a transcription factor, a master regulator that controls the expression of many other genes. This molecular conductor helps orchestrate the development of neural circuits in regions of the brain involved in speech and language processing. This discovery was merely the first thread in a complex genetic tapestry. Researchers began unraveling FOXP2's evolutionary significance by comparing the human version with those found in other animals. The gene exists in many species: mice, birds, even reptiles. But the human version contains specific differences absent in our closest relatives. When scientists compared the amino acid sequence between humans and chimpanzees, they found just 3 differences out of 715 amino acids. 2 of these changes were unique to humans, emerging after our evolutionary line split around 6 million years ago. For a gene so critical to function, this variation was remarkable. Most important genes remain highly conserved across species because any change could be catastrophic. Yet this gene showed human-specific changes coinciding with our species' unique language abilities. Genetic analysis suggested these changes appeared approximately 200,000 years ago and spread rapidly throughout the human population, timing that aligns closely with the emergence of anatomically modern humans and the cultural explosion that followed. The discovery opened new research avenues. Scientists created mice with the human version of FOXP2 to study its effects. These mice showed subtle differences in vocalizations and changes in neural structures involved in motor learning. The KE family's developmental verbal dyspraxia provided concrete evidence of a genetic basis for human language. Their struggle revealed a biological foundation for our most human trait. Through a single mutated gene in one family, scientists gained a window into the biological machinery that makes language possible. This finding challenged long-standing beliefs about language acquisition. For decades, many linguists had argued that language was primarily a cultural phenomenon, something we learn entirely through exposure and socialization. The FOXP2 discovery demonstrated that genetic factors play a crucial role in our ability to acquire language. Without the proper genetic foundation, normal language development simply cannot occur. The identification of FOXP2 showed how a mutation in a single gene could disrupt a complex cognitive ability. It demonstrated that despite language's complexity, aspects of it could be traced to specific biological mechanisms. This didn't mean language was contained within this gene. Rather, the gene participated in building the neural architecture that makes language possible. Perhaps most significantly, the discovery provided the first molecular entry point into understanding the genetic basis of human language. It gave scientists a starting place to investigate the broader genetic networks involved in language processing. Researchers could now explore what genes FOXP2 regulates, what brain structures it helps develop, and how changes in this gene contributed to human evolution. But what exactly happens when this crucial gene doesn't work properly? The KE family showed us the broad outlines, but in the years since this initial discovery, researchers have developed a much more detailed understanding of how FOXP2 mutations disrupt the intricate machinery of human language. When language breaks down, what happens when the genetic foundation for our most human ability breaks down? The FOXP2 gene, when functioning normally, orchestrates the development of neural pathways essential for speech. But when mutations occur, they disrupt this intricate process with profound consequences. People with FOXP2 mutations struggle with speech tasks that most of us perform without conscious thought. Imagine trying to say the word baseball, but being unable to coordinate your tongue, lips, and jaw to produce those sounds consistently. One attempt might yield bashball, the next fadeball, and a third something entirely different. This inconsistency is a hallmark of FOXP2-related speech disorders. The condition caused by FOXP2 mutations has a clinical name, developmental verbal dyspraxia, also known as childhood apraxia of speech. This disorder isn't about understanding language. Individuals with this condition comprehend words and sentences just fine. Their challenge lies in the physical production of speech. The brain knows what to say, but cannot reliably send the precise sequence of commands to the muscles involved in speaking. Children with this condition struggle from their earliest attempts at speech. While typical babies progress from babbling to first words to simple sentences in a predictable sequence, those with FOXP2 mutations show significant delays and atypical patterns. Their speech development follows an entirely different trajectory. The disorder manifests in several characteristic ways. First, affected individuals have difficulty with sequencing sounds. The word temperature might become temperature or temperature. Second, they struggle with prosody, the rhythm, stress, and intonation of speech. Their speech often sounds monotone or has unusual stress patterns. Third, they show groping behaviors, visible struggles as they try to position their articulators correctly. Like a broken orchestra conductor who knows the melody but can't coordinate the musicians, the brain cannot properly sequence the precise movements needed for speech. You might see a child's mouth making several attempts before a sound emerges. What makes this condition particularly challenging is the variability of errors. The child must relearn the motor plan for each word every time they attempt to say it, making traditional speech therapy approaches less effective. FOXP2 mutations affect brain development in specific and measurable ways. Brain imaging studies reveal differences in several key regions that form the neural circuit for speech and language. The basal ganglia, deep brain structures involved in coordinating movement sequences, show altered development. Broca's area, a region in the left frontal lobe critical for speech production, displays abnormal activity patterns during language tasks. The cerebellum, which plays a significant role in timing and motor coordination, shows structural differences. Additionally, the arcuate fasciculus, a bundle of nerve fibers connecting language comprehension areas with speech production regions, appears underdeveloped. Consider the case of a 10-year-old boy with a confirmed FOXP2 mutation. His brain scans revealed significantly reduced gray matter volume in the basal ganglia and abnormal activation patterns in Broca's area during speech tasks. Despite average intelligence and good comprehension, his speech remained largely unintelligible to unfamiliar listeners. When attempting to repeat simple sentences, his brain activity showed chaotic patterns compared to the organized neural firing seen in typical children. These brain differences emerged during early development. FOXP2 acts as a genetic switch, turning other genes on and off at specific times during brain formation. When mutated, it fails to activate the proper developmental cascade, leading to abnormal neural circuitry that persists throughout life. The impacts of FOXP2 mutations extend beyond speech. Many affected individuals also show broader motor coordination difficulties, affecting skills like tying shoelaces, riding a bicycle, or manipulating small objects. This suggests FOXP2 plays a role in motor learning throughout the body. Intriguingly, researchers have noted overlap between FOXP2-related disorders and autism spectrum disorders. While not all individuals with FOXP2 mutations meet the criteria for autism, some share characteristics like repetitive behaviors, social communication difficulties, and restricted interests. This connection hints at FOXP2's role in broader neural networks that support social communication beyond just speech production. The disruption caused by FOXP2 mutations highlights the gene's role as a master regulator. It doesn't directly build speech structures in the brain. Rather, it controls networks of other genes that together construct the neural architecture for language. When this master switch malfunctions, the effects cascade throughout development, affecting multiple systems. Treatment for individuals with FOXP2 mutations focuses on intensive speech therapy using approaches specifically designed for apraxia of speech. These therapies emphasize repetitive practice of movement sequences, multisensory cues, visual, tactile, and auditory feedback, and gradual building of complexity. Progress typically comes slowly and requires significant persistence. Some therapeutic approaches incorporate music and rhythm to bypass the impaired speech motor systems and access alternative neural pathways. Singing, for instance, often remains intact even when speaking is severely impaired, suggesting different neural systems support these activities. Augmentative and alternative communication tools, from simple picture boards to sophisticated speech-generating devices, can provide crucial communication support while speech skills develop. These tools help prevent the social isolation and frustration that often accompanies severe speech disorders. The comprehensive impact of FOXP2 mutations reveals how a single gene can affect multiple interconnected language systems. From the motor aspects of speech production to the cognitive components of grammar processing to the social dimensions of communication, FOXP2 influences the entire language network. This demonstrates that language acquisition depends on both the proper genetic blueprint and appropriate environmental input. What makes this story even more fascinating is that the human version of FOXP2 has unique characteristics not found in other species, with one notable exception. When scientists examined the evolutionary history of this gene, they discovered something unexpected that raises profound questions about human origins. The evolutionary anomaly. How did humans acquire a genetic mutation that no other species on Earth possesses? After scientists identified FOXP2's role in language development, they naturally wanted to understand its evolutionary history. How did this gene change over time to give humans our unique language capabilities? In the early 2000s, researchers began comparing the FOXP2 gene across different species. They extracted DNA from humans, chimpanzees, gorillas, macaques, mice, and other mammals to analyze the genetic code. The scientific team, led by researchers at the Max Planck Institute for Evolutionary Anthropology in Germany, was looking for differences that might explain human language ability. What they discovered shocked the scientific community. Humans possess two specific amino acid substitutions in the FOXP2 protein not found in any other mammals. These changes occur at positions 303 and 325 within the protein structure, where humans have asparagine and serine respectively, while other mammals have threonine and arginine. Those seemingly minor alterations in a protein composed of 715 amino acids, these precise changes represent a fundamental rewiring of our genetic blueprint. The FOXP2 gene shows remarkable stability across mammalian evolution. When comparing mouse and chimpanzee versions, scientists found only 3 amino acid differences despite 70 million years of separate evolution. This extreme conservation indicates the gene serves critical functions that rarely tolerate change. Any significant mutation would likely be harmful or fatal. These human-specific substitutions occur in functionally critical regions. Position 303 sits in the DNA binding domain that controls which genes activate, while position 325 is located in a region that interacts with other proteins. Could these minute changes be the spark that ignited language evolution? Laboratory studies confirm these substitutions alter how FOXP2 regulates other genes. The human version activates and suppresses different target genes compared to the chimpanzee version, affecting the development of neural circuits in brain regions associated with speech. When examining chimpanzees and bonobos, who share 99.8% of their DNA with humans, we find they lack complex language capacity. Much like how a single line of code can transform an entire software program, these tiny FOXP2 modifications appear to have outsized effects on our cognitive abilities. The chimpanzee FOXP2 version matches that of gorillas, indicating stability for 8 to 10 million years since these species shared a common ancestor. This consistency highlights how extraordinary the human changes are. Something in our lineage permitted, or required, these specific mutations to emerge and proliferate. To establish when these changes appeared, scientists constructed an evolutionary timeline based on genetic analysis. By measuring neutral mutations in regions surrounding FOXP2, they estimated the human-specific changes emerged approximately 200,000 years ago, coinciding with the appearance of anatomically modern humans in the fossil record. What's particularly mysterious is how rapidly these mutations spread through the human population. For a mutation to become fixed, present in virtually all members of a species, it typically needs to provide a significant survival advantage. The FOXP2 mutations show signs of a selective sweep, a pattern indicating extremely strong positive selection, spreading much faster than expected by chance alone. The genetic variation around FOXP2 in modern humans shows remarkably little diversity compared to other genome regions, a telltale sign of the selective sweep. It's as if these mutations were so advantageous that they rapidly replaced all alternative versions. Computer models suggest they likely provided at least a 5% reproductive advantage, an enormous benefit in evolutionary terms. The most plausible explanation is that these changes enhance language abilities, providing social and survival advantages. Better communication would improve cooperation in hunting, gathering, and defense. It would enhance teaching and learning, allowing cultural knowledge to accumulate and spread more effectively. Additionally, linguistic ability could signal intelligence and good genes during mate selection. The story took an unexpected turn when researchers examined FOXP2 in Neanderthals. When they sequenced DNA from Neanderthal fossils, they made a startling discovery. Neanderthals had the same 2 amino acid substitutions found in modern humans. This finding contradicted the timeline established by the selective sweep analysis. If these mutations appeared 200,000 years ago in the modern human lineage, how could Neanderthals possess them? The human and Neanderthal lineages diverged approximately 500,000 years ago, well before the estimated emergence of these mutations. The presence of identical FOXP2 mutations in both humans and Neanderthals suggests either that the mutations are much older than initially thought, predating the split between humans and Neanderthals, or that some form of gene flow occurred between the two species. Either explanation complicates our understanding of how and when these crucial genetic changes emerged. These 2 amino acid substitutions represent one of the most striking genetic differences between humans and other primates. In a gene that remained virtually unchanged for tens of millions of years across diverse mammalian species, humans acquired specific changes that altered its function in ways that potentially enabled our unique language capabilities. The evolutionary story of FOXP2 challenges conventional expectations. These mutations appeared with remarkable speed and spread with unusual rapidity, suggesting extraordinary benefits to early humans. Yet their presence in Neanderthals raises questions about their timing and origin. But when exactly did these changes first appear in our evolutionary lineage? The Neanderthal evidence forces us to reconsider the timeline of this critical genetic development. The timing mystery. When exactly did the genetic changes that enabled human speech emerge? Pinpointing this moment has proven challenging for scientists, with surprising discoveries forcing researchers to repeatedly revise their timelines. Geneticists used molecular clock analysis to determine when the human FOXP2 variant first appeared. This approach measures the rate at which neutral mutations accumulate in DNA surrounding the gene, allowing scientists to calculate when specific genetic changes emerged. In 2002, researchers at the Max Planck Institute analyzed FOXP2 in diverse human populations worldwide. They found minimal genetic variation in regions surrounding two key amino acid changes, suggesting a selective sweep, a rapid spread of beneficial mutations through the entire human population. This analysis initially estimated these mutations appeared approximately 200,000 years ago, aligning with fossil evidence for anatomically modern humans in Africa. The timing created a compelling narrative. The human FOXP2 variant coincided with our species' emergence, potentially explaining our cognitive advancement. However, this timeline was disrupted in 2007 when scientists analyzed Neanderthal DNA. Remarkably, Neanderthals possessed identical amino acid substitutions in their FOXP2 gene. This discovery immediately called the previous dating into question. Since humans and Neanderthals diverged approximately 500,000 years ago, these changes must have occurred before their split, much earlier than previously thought. This revelation created a significant puzzle. If the mutations appeared over 500,000 years ago, why did genetic evidence suggest a much more recent selective sweep? What does this mean for our understanding of human evolution? Several explanations emerged. The initial dating methodology might have been flawed, relying on assumptions about mutation rates and population sizes that weren't accurate. More recent analyses suggest the selective sweep might indeed be older than initially calculated. Alternatively, while the mutations themselves might be ancient, they could have undergone a second selective sweep more recently. Perhaps these genetic changes initially provided modest benefits but later combined with other mutations to produce significant advantages in modern humans. A third possibility involves gene flow between humans and Neanderthals. The mutations might have originated in one lineage and crossed into the other through interbreeding. We know modern humans outside Africa carry 1 to 4% Neanderthal DNA, though the pattern of FOXP2 variation doesn't align well with this explanation. Understanding how quickly these mutations spread is equally important. The genetic evidence indicates an extraordinarily rapid spread, suggesting substantial advantages for carriers. For a genetic change to become fixed in a population, it typically must increase reproductive success. In this case, by approximately 5%, which represents a significant evolutionary advantage. Between 200,000 and 50,000 years ago, humans underwent an explosion in cultural complexity. They developed sophisticated stone tools, created art, established trade networks, and expanded into new environments, marking what anthropologists call the transition to behavioral modernity. Did FOXP2 mutations enable this cultural explosion? The timing suggests a connection, but if these mutations are indeed much older, we must explain why they didn't produce similar advances in earlier human populations or in Neanderthals. One explanation is that FOXP2 alone wasn't sufficient. These mutations may have created potential for advanced language, but additional genetic or cultural developments were needed to fully realize this potential. Language likely requires multiple genetic changes working together, with FOXP2 being just one component. Other genes involved in language show evidence of recent selection. For example, NOVA1, which helps regulate brain development, has a human-specific variant absent in Neanderthals. This mutation emerged approximately 250,000 to 300,000 years ago. When researchers introduced this human version into mice, it altered their vocalizations, suggesting multiple genes underwent selection at different points in human evolution. If Neanderthals possess the same FOXP2 variant as modern humans, do they also have language? Archaeological evidence indicates Neanderthals had complex behaviors and may have had anatomical structures needed for speech. Yet they never developed the rich cultural traditions seen in modern human societies. Recent studies add another layer of complexity, suggesting that while Neanderthals had the same amino acid substitutions, they may have had differences in regulatory regions controlling gene expression. These differences could have affected gene function despite identical protein structure. The debate highlights how much remains unknown about the genetic foundations of human language. The FOXP2 story demonstrates both the power of genetic analysis to illuminate our evolutionary past and the challenges in interpreting this evidence. What we can confidently say is that the human variant of FOXP2 emerged at a crucial point in our evolutionary history. Whether these changes appeared 200,000 or 500,000 years ago, they coincided with a period when our ancestors were developing the cognitive and social capacities that distinguish humans from all other species. The FOXP2 mutations appeared during humans' unprecedented cognitive advancement. Their timing likely played a causal role in the emergence of modern human cognition and culture. Even if they appeared earlier than initially thought, their impact became most evident during the transition to behavioral modernity. But how exactly does FOXP2 enable language? What biological mechanisms translate these genetic changes into the remarkable ability to speak and understand complex language? The answer lies in how does a single gene enable the complex neural circuitry to speak and understand language? The answer lies in understanding FOXP2 not as a language gene, but as a master regulator that orchestrates the development of neural circuits essential for human communication. FOXP2 serves as the maestro of neural development, conducting a symphony that extends well beyond language processing. As a transcription factor, it binds to specific DNA sequences, controlling hundreds of other genes during brain development and shaping the neural architecture that makes language possible. The human version of FOXP2 differs from the chimpanzee version in just 2 amino acids, yet these tiny changes significantly alter how the protein interacts with DNA. This human variant regulates approximately 300 to 400 gene promoters within the genome, controlling genes crucial for brain development, neural connectivity, and motor learning. Evidence from mouse experiments illuminates FOXP2's profound impact. Researchers created humanized mice by replacing their FOXP2 with the human version. These mice developed longer dendrites in striatal neurons and showed increased synaptic plasticity. Their vocalizations changed, producing different patterns of ultrasonic squeaks. Most significantly, they demonstrated enhanced procedural learning abilities, mastering maze patterns faster and better automatizing repeated behaviors, capabilities directly relevant to human language acquisition. FOXP2 doesn't directly create language. It builds the biological systems that make language possible. During critical developmental periods, it influences how neurons grow, connect, and form circuits in brain regions crucial for speech and language processing. The basal ganglia represent one primary site of FOXP2 action. These deep brain structures coordinate the precise movements required for speech, orchestrating over 100 muscles in your lips, tongue, jaw, and larynx to produce rapid, accurate sound sequences. FOXP2 expresses at high levels in the developing striatum, a key basal ganglia region, shaping connections that enable rapid sequencing of fine motor movements. When FOXP2 mutations disrupt these circuits, individuals struggle with speech coordination. The cerebellum, another FOXP2 target, contributes both to movement coordination and cognitive functions including language processing. FOXP2 expression here establishes neural circuits essential for the timing and precision of speech movements. Beyond motor regions, FOXP2 influences cerebral cortex areas central to language. It shapes Broca's area in the left frontal lobe, crucial for speech production and grammar processing, while affecting circuits connecting to Wernicke's area for language comprehension. Perhaps FOXP2's most powerful influence lies in synaptic plasticity, the neural flexibility that allows connections to strengthen or weaken through experience. Language acquisition depends on this adaptability as we learn and refine speech through practice. FOXP2 regulates genes involved in plasticity, particularly in circuits connecting the cortex, basal ganglia, and thalamus, pathways critical for procedural learning and speech motor control. This connection to procedural learning holds special significance. Language acquisition involves both declarative components, vocabulary and explicit grammar rules, and procedural components, internalizing patterns through practice until automatic. FOXP2 appears particularly vital for these procedural aspects, the unconscious acquisition of speech patterns and grammatical structures through repeated exposure. FOXP2's influence extends beyond the brain to organs including the lungs and gut, explaining why some individuals with mutations show mild abnormalities outside speech and language domains. As a transcription factor, FOXP2 orchestrates complex developmental processes by regulating numerous genes involved in neural migration, axon guidance, and synapse formation. Specific genes under its control include CNTNAP2, affecting neuron migration and connection, SRPX2, influencing synapse formation in language-related regions, and FOXP1, another transcription factor working alongside FOXP2 in neural development. This genetic network explains FOXP2's profound effects on language ability. The protein coordinates a symphony of genetic activities that collectively build the neural architecture supporting speech and language. When we speak, this intricate system allows words to flow with precision and meaning, enabling the uniquely human capacity for complex communication. The FOXP2 story reveals language as a sophisticated biological trait depending not on a single gene but on an elaborate genetic network building and maintaining neural circuits responsible for various language aspects. FOXP2 conducts this genetic orchestra, creating the biological foundation for human language. This understanding helps explain the diversity of language disorders. Depending on which part of this genetic network is disrupted, individuals might struggle primarily with speech production, grammar comprehension, vocabulary learning, or reading ability. FOXP2 mutations typically affect speech motor control, reflecting the gene's strong influence on related circuits. FOXP2 transcends its designation as merely a language gene. Evidence points to its involvement in broader cognitive and developmental processes. Through its far-reaching influence, this remarkable gene helps create the biological foundation for one of humanity's defining abilities, the power to communicate complex thoughts through language. Beyond language, is FOXP2 really just a language gene, or is there more to the story? When FOXP2 was first linked to speech disorders in the late 1990s, media outlets quickly labeled it the language gene, creating the impression that a single gene was responsible for human language. This simplification caught on, but it misrepresents the true nature of this remarkable gene. The reality is far more complex. FOXP2 functions like a master craftsman's multi-tool, seemingly designed for one purpose but containing specialized components for diverse applications. Research over the past 2 decades has revealed that FOXP2 influences multiple biological systems beyond speech and language processing. FOXP2's versatility extends to several key areas: motor learning and coordination. Studies in mice show FOXP2 is crucial for learning complex motor sequences unrelated to vocalization. Mice with altered FOXP2 struggle to learn rotarod running, a task requiring precise coordination. This explains why people with FOXP2 mutations often show broader coordination difficulties like trouble with handwriting, tying shoelaces, or manipulating small objects. Neural development. FOXP2 regulates neuronal growth and connectivity in the cerebellum, thalamus, and cortex, regions involved in diverse functions from sensory processing to executive control. Non-neural development. During embryonic development, FOXP2 appears in the lungs, heart, and digestive system, contributing to tissue formation in these organs. This explains why some individuals with FOXP2 mutations show minor physical abnormalities beyond their speech and language deficits. The evidence for FOXP2's diverse functions comes from studies across the animal kingdom. In songbirds like zebra finches, FOXP2 enables proper song learning. When researchers reduce FOXP2 expression in a brain region called Area X, young birds produce incomplete or imprecise vocalizations, similar to speech problems in humans with FOXP2 mutations. However, the zebra finch version of FOXP2 differs significantly from the human version, suggesting independent evolution for vocal learning in different species. Bats, which navigate using echolocation, have unique FOXP2 variants that differ from other mammals, possibly related to the precise vocal control needed for echolocation pulses rather than communication. FOXP2 also responds to environmental context. In songbirds, FOXP2 expression changes based on social context, differing between courtship songs directed at females and practice songs sung alone. This suggests FOXP2 links communication skills with social behavior more broadly. Having to reframe FOXP2 as more than just a language gene challenges our desire for simple explanations. We naturally seek straightforward narratives, a gene for this, a gene for that, but biological reality rarely obliges. The complexity of FOXP2 reminds us that even our most fundamental human capacities emerge from intricate genetic networks rather than single genetic switches. This broader perspective helps explain why language evolution is so complex. Language didn't evolve through changes to a single gene. Instead, it emerged through modifications to genes like FOXP2 that were already involved in motor control, learning, and social communication. Human language co-opted and refined existing biological systems rather than creating entirely new ones. FOXP2 works alongside many other genes to build the neural architecture supporting language and other cognitive abilities. These partner genes include CNTNAP2, which affects neural migration, FOXP1, a related transcription factor, and various genes involved in synaptic plasticity and neural development. Understanding FOXP2 as a multifunction regulator has important implications. Clinically, it suggests treatments for language disorders must consider effects beyond just speech circuits. Evolutionarily, it explains how language could evolve by repurposing existing genetic machinery rather than requiring entirely new innovations. FOXP2 exemplifies how evolution works with available materials. Rather than creating a dedicated language gene, natural selection modified an existing gene that already influenced motor learning and neural development. Through specific mutations, this gene adapted to support human language while maintaining its other functions. The story of FOXP2 teaches us that genes rarely have single functions. Like most genes, FOXP2 participates in numerous biological processes throughout development. Its role in language represents just one facet of its broader significance. So while FOXP2 contributes significantly to human language abilities, calling it the language gene oversimplifies its nature. It's better understood as a versatile regulator that helps orchestrate multiple biological systems. Language emerges not from a single gene but from many genes working together to build the remarkable architecture enabling human communication. This broader understanding raises intriguing questions about how FOXP2 reshapes the brain to enable language. What specific neural differences can be directly attributed to FOXP2's influence? The answer lies in examining how this gene transforms brain structure and connectivity. The rewired brain. How did FOXP2 transform the human brain to enable language? When scientists compare human brains to those of other primates, they find striking structural differences, particularly in regions associated with language processing. These differences didn't emerge by chance. They reflect genetic changes that rewired the language circuitry of our brain during human evolution. The human brain differs from chimpanzee and gorilla brains in several key ways. While overall brain size represents one obvious difference, with human brains roughly 3 times larger than chimpanzee brains, the more significant differences involve specific brain regions and their interconnections. These specialized adaptations support our unique language capabilities. The language processing regions of the cerebral cortex show prominent differences. Humans possess enlarged and more complex versions of Broca's and Wernicke's regions. Broca's area in the left frontal lobe controls speech production and grammar processing, while Wernicke's area in the left temporal lobe handles language comprehension. Although primates have anatomically similar regions, the human versions display significant expansions and specializations. FOXP2 directly influences the development of these language-critical brain regions. During embryonic development, FOXP2 is expressed at high levels in the emerging neural circuits that form these specialized language areas. The gene functions as a master switch, regulating hundreds of other genes that guide how neurons grow, connect, and form circuits. The human version of FOXP2, with its unique amino acid changes, alters this regulatory network, creating different patterns of neural development in these critical regions. A particularly important feature of the human brain is its pronounced asymmetry in language-related structures. Language functions are predominantly lateralized to the left hemisphere in most people. This hemispheric specialization allows for more efficient neural processing. Research indicates that FOXP2 influences this lateralization process during brain development. Studies of individuals with FOXP2 mutations reveal altered patterns of brain asymmetry, particularly in language-related regions. This suggests the gene helps establish the left hemisphere dominance typical of human language processing. Without proper FOXP2 function, this asymmetry doesn't develop correctly, contributing to language difficulties. FOXP2 also shapes the connections between different brain regions involved in language processing. Human language requires seamless integration between areas that process auditory information, those that plan speech movements, and those that handle grammar and meaning. Like an intricate transportation network, these neural pathways must connect precisely the right regions with optimal efficiency. One crucial connection strengthened in the human brain links motor regions controlling the mouth, tongue, and larynx with auditory processing areas. This motor auditory circuit creates a feedback loop essential for speech learning. When you speak, your brain constantly monitors the sounds you produce. compares them to the intended sounds, and makes rapid adjustments to your vocal apparatus. This feedback mechanism allows you to learn new words, correct pronunciation errors, and adapt your speech to different contexts. FOXP2 influences the development of these feedback circuits by regulating genes involved in axon guidance, the process by which developing neurons extend connections to their targets. The human FOXP2 variant alters the expression of genes that guide these growing connections, enhancing the pathways between motor and auditory regions. Research on mice with the human version of FOXP2 supports this idea. These humanized mice show increased connectivity in circuits linking the cortex with the striatum and thalamus, pathways critical for motor learning and sensory integration. While mice don't speak, these altered neural circuits affect their vocalizations and learning abilities, suggesting similar changes in humans could support speech acquisition. Another key connection influenced by FOXP2 is the arcuate fasciculus, a neural superhighway linking Broca's area with Wernicke's area. This connection allows information to flow between brain regions responsible for language comprehension and those involved in speech production. In humans, this connection is significantly larger and more developed than in other primates, enabling the translation of thoughts into speech and the processing of heard language into meaning. FOXP2 also affects synaptic plasticity, the ability of neural connections to strengthen or weaken based on experience. Language acquisition depends heavily on this plasticity, as we refine our neural circuits based on language exposure. The human FOXP2 variant regulates genes involved in synapse formation, enhancing the brain's capacity to learn language. Brain imaging studies reveal the consequences of these neural changes. When people with normal FOXP2 function process language, they show coordinated activity across specialized brain networks. Individuals with FOXP2 mutations display atypical activation patterns and altered connectivity between language regions. The gene shows high expression during critical periods when language circuits are forming, particularly during mid-fetal development and early childhood. These periods represent windows of opportunity when the brain is especially receptive to language input. FOXP2 regulates neuronal migration, the process by which newly formed neurons travel to their proper locations in the developing brain. This ensures neurons reach the correct brain regions and form appropriate connections. Disruptions to this process can alter the structure of language-related brain areas. The brain changes influenced by FOXP2 mirror the genetic changes that distinguish human FOXP2 from other versions. The two amino acid substitutions unique to humans alter how the FOXP2 protein interacts with DNA, changing which target genes it activates or suppresses. These molecular changes translate into altered neural development, producing the specialized brain circuits that enable human language. But one of the most surprising discoveries about FOXP2 involves our evolutionary cousins, the Neanderthals. These extinct hominids shared a common ancestor with modern humans approximately 500,000 years ago and survived until about 40,000 years ago. When scientists analyzed DNA extracted from Neanderthal fossils, they found something unexpected that raises profound questions about human uniqueness and the origins of language. The Neanderthal Puzzle. Why do Neanderthals share our unique version of FOXP2 when no other species does? In 2007, scientists made a discovery that shocked the evolutionary biology community. After successfully extracting and analyzing DNA from Neanderthal fossils, researchers found something unexpected. Neanderthals possess the exact same version of FOXP2 as modern humans, including the two amino acid changes previously thought to be unique to our species. This finding completely upended our timeline of language evolution, noted Dr. Svante Pääbo, one of the researchers who led the breakthrough study. The Neanderthal Puzzle challenges what we thought made us unique and forces us to reconsider the very foundations of human exceptionalism. Until this discovery, many researchers had assumed the human-specific changes to FOXP2 had emerged relatively recently, perhaps coinciding with the appearance of anatomically modern humans around 200,000 years ago. The presence of these same genetic changes in Neanderthals overturned this timeline completely. Humans and Neanderthals diverged from a common ancestor approximately 500,000 years ago. If Neanderthals carried the same FOXP2 variant as modern humans, these mutations must have occurred before this split, more than twice as far back in time as previously thought. This dramatically changed our understanding of when these critical genetic changes first appeared. The genetic evidence is compelling. When researchers analyzed FOXP2 sequences from several Neanderthal specimens from different locations and time periods, they consistently found the same pattern. At positions 911 and 977 in exon 7 of the FOXP2 gene, the exact locations of the human-specific changes, Neanderthals had identical mutations to modern humans. This shared genetic heritage suggests something remarkable. The mutations that potentially enabled modern human language emerged not in our species, but in the common ancestor we shared with Neanderthals. These changes occurred in a hominin species that predated both modern humans and Neanderthals, somewhere between 500,000 and 800,000 years ago. This finding complicates the evolutionary narrative substantially. If these genetic changes are indeed crucial for language development, as studies of the K family suggest, then Neanderthals potentially possess the genetic foundation for complex language. This challenges the view that sophisticated language was unique to Homo sapiens and a key factor in our species' success. The implications extend beyond genetics into how we interpret archaeological evidence of Neanderthal cognitive abilities. For decades, Neanderthals were portrayed as cognitively inferior to modern humans. But this genetic evidence, combined with growing archaeological discoveries, paints a very different picture. Neanderthals created complex tools requiring planning and foresight. They controlled fire, built shelters, and buried their dead, sometimes with grave goods suggesting symbolic thinking. They created jewelry from eagle talons and seashells, used pigments like ochre likely for body decoration, and produced cave art in Spain that predates the arrival of modern humans in Europe. All these behaviors suggest cognitive complexity compatible with some form of language. The presence of the human FOXP2 variant adds biological support to this archaeological evidence, suggesting Neanderthals may have possessed communication abilities far more sophisticated than previously thought. This convergence of genetic and archaeological evidence creates a compelling case that Neanderthals likely possessed significant language ability. They had the genetic foundations, the cognitive complexity, and the necessary physical attributes for speech. But this raises profound questions about human uniqueness. If Neanderthals shared our language gene and potentially had some form of language, what truly separates our species from them? Why did modern humans ultimately thrive while Neanderthals went extinct? The shared FOXP2 variant suggests that language alone may not explain the different fates of our two species. Perhaps the difference wasn't the presence or absence of language, but rather its complexity. Modern humans might have developed more sophisticated linguistic systems, even with the same genetic foundations. Or perhaps the difference lay in how language was used for more complex social organization, more efficient knowledge transmission, or more flexible adaptation to changing environments. Another possibility is that FOXP2, while important, is just one piece of a larger genetic puzzle. Modern humans may have developed additional genetic adaptations that enhanced our communicative abilities beyond what Neanderthals possessed. Recent research has identified other genes involved in language and cognition that show differences between modern humans and Neanderthals. The shared FOXP2 variant also raises questions about the earliest members of the Homo genus. If this mutation occurred before the human Neanderthal split, did even earlier species like Homo heidelbergensis also possess it? Could the foundations for language stretch even further back in our evolutionary history? This discovery challenges us to reconsider not just Neanderthals but our entire understanding of human evolution. Language may have evolved gradually across multiple hominin species over hundreds of thousands of years. The genetic foundations for this quintessentially human trait have deeper roots than we previously recognized. The Neanderthal FOXP2 finding represents one of the most significant challenges to our understanding of human uniqueness. It suggests that what we consider distinctly human may in fact be shared with our extinct relatives, forcing us to reconsider what truly makes our species special. But if the same FOXP2 mutations appeared in a common ancestor of humans and Neanderthals more than 500,000 years ago, this raises another profound question. How likely is it that such specific genetic changes would occur randomly? The precise nature of these mutations, their apparent importance for language, and their timing in evolutionary history have led some scientists to question beyond random natural selection. Could random mutation really explain the emergence of such a complex and critical genetic change? The human FOXP2 gene contains two specific amino acid substitutions not found in other primates except Neanderthals. These changes, while small in number, have significant effects on brain development and potentially enabled human language capabilities. But the precision and timing of these mutations raise important questions about their origins. When geneticists analyze mutation rates in human evolution, they find that our genome typically accumulates between 40 and 80 new mutations per generation across our 3 billion base pairs of DNA. Most mutations occur randomly, with the vast majority having no effect or being slightly harmful. Beneficial mutations providing a survival advantage are extremely rare. The FOXP2 mutations stand out because of their remarkable specificity. The human version differs from the chimpanzee version at exactly 2 amino acid positions 303 and 325. At position 303, a change from ACA to AAC transformed the amino acid from threonine to asparagine. At position 325, a change from AGA to AGT transformed arginine to serine. These genetic changes represent a biological lottery of extraordinary odds. Imagine needing to roll 2 specific numbers on 2 dice, but with billions of possible faces rather than just 6. The mutations had to occur at precise positions within the gene, required particular nucleotide changes, and both had to happen in the same genetic lineage. Geneticists estimate the probability of these exact mutations occurring randomly and spreading throughout a population is vanishingly small. For these mutations to become universal in humans, they needed to provide immediate benefits. Natural selection only preserves changes that enhance survival and reproduction. If the FOXP2 mutations hadn't offered significant advantages, they would likely have disappeared rather than becoming fixed in our species. Evolutionary biologists suggest these mutations conferred substantial benefits to early humans. They may have enhanced communication, improved coordination during hunting, enabled more effective teaching of survival skills, or strengthened social bonds. These advantages could have increased survival rates significantly enough to overcome the statistical improbability. The conventional explanation relies on a selective sweep, where a beneficial mutation spreads rapidly through a population. Evidence comes from the pattern of genetic variation surrounding FOXP2. Humans show unusually low diversity in this region compared to other genome parts, suggesting strong selection pressure favored a specific variant quickly. Scientists estimate these mutations likely provided at least a 5% reproductive advantage to carriers, substantial in evolutionary terms. Over generations, this advantage would allow the mutations to become universal despite their initial rarity. Alternative explanations include genetic hitchhiking, where the FOXP2 mutations occurred alongside other beneficial genetic changes, and the entire package was selected for. This could explain how such specific mutations became established despite their low independent probability. Some researchers propose more controversial explanations involving directed mutation or non-random evolutionary pressure, suggesting certain genome regions might experience higher mutation rates in response to environmental pressures. The mathematical improbability becomes even more striking considering these identical mutations appear in Neanderthals. For both hominin lineages to independently develop identical changes through random processes would be extraordinarily unlikely. The shared FOXP2 variant suggests these changes occurred in a common ancestor before humans and Neanderthals diverged, at least 500,000 years ago. This extended timeline provides more generations for the mutations to occur and spread, somewhat reducing the statistical problem. However, it creates a new puzzle. If these mutations were so beneficial and appeared so long ago, why didn't they produce language capabilities in earlier hominin species? One possibility is that FOXP2 provided necessary but insufficient conditions for language. Additional genetic changes may have been required, occurring only in the Homo sapiens lineage after separating from Neanderthals. Cultural and environmental factors might also have played crucial roles beyond genetics alone. Laboratory studies amplify these questions by showing that changing just these 2 amino acids in the mouse version of FOXP2 to match the human version alters neural development and vocalization patterns. Such profound effects from such specific changes heighten the mystery of their origin. FOXP2 represents just one of many genes showing evidence of accelerated evolution in humans. Genes involved in brain development, immune function, and metabolism all show signs of strong positive selection in our evolutionary history, suggesting powerful selective pressures acted on multiple aspects of human biology simultaneously. The statistical rarity of these precise genetic changes touches on fundamental questions about chance versus direction in evolution. While science can calculate probabilities and document genetic changes, interpreting their significance involves larger questions about human origins. Whether through an unlikely stroke of genetic fortune, strong selective pressures, or mechanisms not yet fully understood, these mutations represent a pivotal moment in our evolutionary history that potentially enabled human language. The remarkable precision of these changes continues to challenge our understanding of how such statistically improbable events could have occurred through conventional evolutionary mechanisms. Alternative origins. If not random natural selection, what could explain the sudden appearance of the human FOXP2 variant? While mainstream science offers well-established mechanisms to explain genetic innovations, the specific changes in FOXP2 present unique challenges that have prompted consideration of alternative explanations. Conventional evolutionary theory provides several mechanisms that could account for the FOXP2 mutations. Strong selective pressure, enhanced communication abilities could have improved hunting coordination, knowledge transmission, and social bonding in challenging environments. Sexual selection. Individuals with better communication skills may have attracted more mates, causing language-enhancing mutations to spread quickly. Genetic drift. This random process can cause certain genetic variants to become fixed in a population through chance alone, especially in small, isolated groups experiencing population bottlenecks. These conventional explanations face challenges when applied to the FOXP2 mutations. The specific changes occurred at precise locations within the gene and spread with remarkable speed. The exact same mutations appear in Neanderthals, pushing their origin back at least 500,000 years. This timeline creates puzzles about why these mutations, if so advantageous for language, didn't produce similar cognitive leaps in earlier hominin species. The two amino acid substitutions that distinguish the human FOXP2 protein from that of other primates appeared with remarkable speed, which is difficult to reconcile with the typical timescales of evolutionary change. Note researchers who have studied the gene's evolution. The selective sweep that spread this gene through the human population also occurred rapidly, suggesting a strong selective advantage. But what if chance isn't the whole story? Beyond conventional mechanisms, more controversial explanations have emerged. Horizontal gene transfer, the movement of genetic material between organisms through means other than parent-to-offspring inheritance, offers one possibility. While common in bacteria, this process rarely occurs in mammals. However, unique circumstances might have allowed genetic material to transfer between species, introducing novel variants into the human lineage. Viruses provide another potential vector for genetic changes. Retroviruses can insert their genetic material into host DNA, occasionally introducing new genetic elements that become fixed in a population. Some scientists suggest viral introduction as a mechanism for rapid genetic changes in human evolution, though direct evidence for viral involvement in FOXP2's evolution remains lacking. More speculative theories propose directed panspermia, the idea that life on Earth was seeded or influenced by extraterrestrial sources. Under this framework, the specific FOXP2 mutations might represent intentional genetic modifications introduced by advanced beings. Proponents point to the statistical improbability of such precise mutations occurring randomly and their outsized effect on human cognitive evolution. The ancient astronaut theory extends this concept, suggesting deliberate genetic engineering by advanced extraterrestrial beings. Proponents argue that the sudden appearance of language capability, coinciding with the emergence of modern human behavior approximately 200,000 years ago, represents a significant leap in evolution. The rapid transition in the FOXP2 protein, with 2 amino acid changes separating humans from other primates, suggests that this protein evolved recently on the evolutionary timescale, perhaps even coinciding with the first anatomically modern humans 200,000 years ago, leading to their survival and success, notes one research summary on the topic. Mainstream science responds to these alternative theories with skepticism. For horizontal gene transfer, scientists note the absence of a plausible mechanism between distantly related species. For viral introduction, they highlight the lack of viral sequences associated with FOXP2 in the human genome. As for directed panspermia and ancient astronaut theories, scientists emphasize that these explanations lack testable predictions and violate the principle of parsimony. The idea that simpler explanations should be preferred when both explain the data equally well. Proponents of alternative theories, such as those related to directed panspermia, argue that the complexity and specificity of the FOXP2 changes are unlikely to have arisen through random mutation and natural selection alone, notes one assessment. However, these arguments often rely on statistical improbabilities and do not provide a testable mechanism for how such a directed process could have occurred, making them difficult to validate scientifically. Scientists also point out that statistical improbability alone doesn't invalidate natural selection. Evolutionary history contains many examples of unlikely but beneficial mutations that spread through populations. All explanations face unanswered questions. The incomplete fossil record and limitations in ancient DNA recovery complicate efforts to resolve these questions definitively. We cannot directly observe the evolutionary process that produced the human FOXP2 variant, forcing scientists to rely on indirect evidence and inference. While mainstream science favors natural selection as the primary mechanism behind FOXP2's evolution, the specific details remain incompletely explained. The exact nature of this advantage and the specific environmental pressures that drove the evolution of FOXP2 are still not fully understood, acknowledge researchers in the field. This scientific humility is important. It acknowledges both the explanatory power of established evolutionary mechanisms and the genuine mysteries that remain in understanding human cognitive evolution. The origin of the human FOXP2 variant ultimately connects to larger questions about human uniqueness. Whatever its source, whether conventional evolution, rare genetic events, or something more exotic, this gene participated in creating the neural architecture that enables human language. Through this capacity for complex communication, humans developed unprecedented cultural capabilities, transforming our relationship with our environment and with each other. The FOXP2 gene stands as one of evolution's most fascinating puzzles, a genetic change that transformed our species and enabled language, yet appeared with unusual precision. These two specific amino acid substitutions created ripples across human brain development, rewiring neural circuits essential for speech and complex communication. The presence of identical mutations in Neanderthals challenges our understanding of language origins and human uniqueness. This shared genetic heritage suggests language capabilities have deeper evolutionary roots than previously thought. This spark that ignited modern language formed the foundation for human civilization. As research advances, what other mysteries of our evolution will FOXP2's unfolding story illuminate?